Hello everyone, we are Team GXU China. It's my pleasure to introduce our team's project, Microbial Production of Curcumin as a mildew-proof biopigment material. Our presentation will include project overview, system design, human practices, and future perspectives. Let's go from our project overview. In the 1980s, the World Health Organization proposed safety standards for students and children's painting pigments. The table on the left shows 8 major metals limiting value in pigments published by European Union. However, the problem of excessive heavy metals in pigments is still threatening human health. Besides, the problem of canvas being susceptible to moisture and mold is also troubling people in the field of art. Since the heavy metals in traditional pigments mainly come from coloring materials such as lead chrome yellow, carrying heavy metal impurities is inevitable. Hence, we decide to replace traditional mineral pigments with biopigments which have mildew-proof functions. We found that curcumin is a great choice. Curcumin has attracted worldwide attention because of its abundant advantages. Its strong colorability and mildew-proof functions can help people solve 2 problems above. But the method of extracting curcumin from plants is squeezing and dissolving, which consumes a lot of energy, and the output is far from meeting the market demand. So we imagine using Escherichia coli to produce curcumin. Compared with traditional production methods, this is a more efficient, eco-friendly, and controllable way. Next, let my partner to introduce our work of experiment.
As shown in the slide, the synthesis of curcumin includes 4 essential enzymes: 4Cl, ACC, DCS, and CURS. So our strategy is to generate engineering ecology expressing these enzymes to make curcumin production. By taking advantage of the endogenous indodiacetyl coenzyme 8 in E. coli, we can realize the synthesis of curcumin in the engineering E. coli by supplying ferulic acid as the initial substrate. To express 4 enzymes at the same time, we intended to create 2 constructs. To clone the enzyme genes, we extract RNA from Arabidoxyderianor and perform revised transcription to generate cDNA. cDNA was used as a template to clone ACC and 4Cl genes. The plasmid PAD2A first, enzyme-linked ACC, then enzyme-linked 4Cl. In the same way, we cloned DCS genes from turmeric and CURS from fresh ginger, and then fuse the 2 genes to construct a PGES MCM, DCS, CURS plasmid. We design 3 parts as our code engineering work. Next is a summary of the experimental result. Firstly, agarose gel image demonstrated that we obtained a touchy ACC gene fragment and the plasmid site was correct, indicating the ACC gene had been successfully constructed into PAD2A. Similarly, the verification result of plasmid site and PCR of gene fragment indicated that ACC4-cell expression system had been successfully constructed. At the meantime, we were working on building PGES MCM, DCS, CURS expression system. Result shows that the key rate limiting enzymes, DCS and CURS, has been constructed successfully. Then we made a resistance screening test. It demonstrated that the engineering strain was successfully obtained. At the same time, to understand the physical and chemical properties of curcumin better and the difference between chemical synthesized curcumin and natural curcumin, we performed mass spectrometry analysis of curcumin, which could carry out the follow-up fermentation experiment. The 2 pictures below shows that the extract of turmeric stem surely contained curcumin.
This is my presentation for our experiment. Next, my partner will introduce the hardware section.
This project has designed a temperature and humidity Internet of Things monitoring system based on STM32 single-chip microcomputer, which is divided into 2 parts, the sending end and the receiving end. OLED displays. DHP11 collects temperature and humidity, and the Wi-Fi module delivers the information to the server. The receiving end is displayed in the form of a web page. The web page interface includes the address port of the server, the subject of the subscription, the data read, the relevant commands sent, and so on. After the data is processed by the MCU, data uploaded to the server via the MQTT protocol, and related data can be sent and received by subscribing to the corresponding topics on the website.
In this section, we used polynomial fitting in machine learning to process curcumin decomposition data and predicted the changes in concentration over time. Then we used the same biology in MATLAB to construct an ordinary differential equation to obtain the synchronization law between the concentration of ferulic acid and the concentration of curcumin. Firstly, we constructed a mathematical expression for polynomial fitting to find the time under different dimensions, expanded our data volume, used Python to fit the curve, found the optimum number of dimensions, and obtained the final analytical expression. We defined a metabolic pathway diagram, set internal parameters, verified the correctness of the model, and finally exported it. The reaction rate defined in the model can be determined by consulting literature. The model can well provide the specific concentration of curcumin when the concentration of ferulic acid changes synchronously with the final concentration of curcumin. After integration, the specific concentration of curcumin can also be obtained. In the section of bioinformatics, we first constructed a metabolic pathway map of engineering bacteria, and then used computational biology techniques to construct the structure of the fusion protein, analyzing its stability, interactions, and active centers. We utilized the metabolic pathway in KEGG to better demonstrate the curcumin production metabolic process of the engineered bacteria we constructed, and supplemented the metabolic pathway of MAP 00945. We first fused CURS and DCS, added a linker composed of 3 residues between the 2 protein sequences, inserted it into the plasmid, expressed the fusion protein, and used the AlphaFold2 to predict the 3-dimensional structure of the fusion protein. As shown in the figure, it can be seen that the PLDDT values of most regions of the obtained model are above 90, indicating that the obtained model has high quality. AlphaFold2 predicted the fusion protein model and compared it with the crystal structures of DCS and CURS, resulting in RMSD of 0.7 and 0.501, respectively. This indicates that the 2 parts of the fusion protein have high consistency with the corresponding crystal structures. From the perspective of structural biology, the conformation of the complex formed by the 2 parts of the fusion protein is reasonable. Therefore, it is speculated that there is interaction between the 2 protein monomers used to construct the fusion protein. The study further analyzed and visualized the interaction between DCS and the CURS, and the results showed that there were a large number of hydrogen bonds between residues on the interaction surface of the 2 proteins, and some residues also formed salt bridges. The study further validated the interaction between 2 unilateral complexes using molecular dynamic simulation. The molecular dynamic simulation of DCS and the CURS complex at 15 nanosecond was carried out. This further proved that the complex structure of the 2 is very stable and there is interaction between the 2 proteins. Based on the molecular dynamic simulation results of 15 nanosecond, the stability of various parts of the complex was further evaluated. To investigate the catalytic mechanism of the constructed fusion protein on the substrate, machine learning were used to perform pocket prediction on the fusion protein model. Autodoc VINA was used to pair the corresponding 2 sets of substrate molecules into the corresponding pockets of the fusion protein, obtaining the predicted complex between the substrate molecules and the fusion protein. Based on the obtaining docking results, the study analyzed the possible interaction mechanisms between the fusion protein and the various substrates. This provides theoretical guidance for the further design and modification of fusion proteins. Then, let another team member to introduce our human practices work.
The goal of our entire HP is to exhibit the advantages and charm of synthetic biology to the public by illustrating our project that creates curcumin synthesis by engineering equally. We hoped that the public would start to understand the concept of synthetic biology, be inspired by our curcumin project, and ultimately get involved in the synthetic biology world. We will introduce the HP project into 3 parts: the integrated HP, collaboration, and public education. At the beginning of the entire HP process, we conducted a few interviews with pigment store owners and customers, trying to understand the current pigment market situation to see if the curcumin is in demand. We found that generally people cared more about the pigment price and the performance, and they provided support for our project. Furthermore, we have released a questionnaire on the purchasing situation of pigments and collected 152 feedback from people in the art field. They are more concerned about factors such as the price, color rendering, and order of pigments. Then we attempted to create several generations of curcumin oil painting sticks based on the formula we consulted, continuously improving their hardness and color rendering, and changing their acidity and alkalinity to change their color. The resulting oil painting sticks basically met the market oil painting stick standards. These are works created by our own oil painting sticks. We have met the coloring requirements and further expanded the dyeing potential of curcumin. We have consulted the traditional Chinese dyeing method in ancient times, the water painting method for dyeing. These are works created by our school students through the experience of water painting dyeing. Next is the collaboration part. First, we have learned a lot from 10th CCIC conference. We and other 7 IGM teams nationwide have finally reached an agreement to hold Sing by Rainbow for Children and Sing by Our Story. The 8 IGM teams draw a series of synthetic biology comics, brochures in different areas. Furthermore, we discussed our project with CAU China and Nanjing BioX team in person, and we agreed to support each other on the projects. Then we exchanged ideas with Nanjing BioX team, which shared the similar theme with us. Apart from offline sections, we also talked about some details and prospects online, which helped us a lot. Then is the education part. The purpose of Sing by Rainbow is to bring colors to children with special disabilities. We went to the Qihang Special Education Center to have a drawing class with these kids. During the class, kids drew colorless picture books designed by us, then happily interacted with the mascot. These are works made by them. The president of the Qihang Special Education Center thanked us for carrying out this project so that they felt these children have not been neglected. Inspired by the popular chess and card game of China, Monopoly, we produced the synthetic biology version of Monopoly game. We also innovatively produced the Brave Challenge mode, which adds more fun and challenges for players. For Symbio Carnival, we invited many students from different grades and colleges to our carnival named Micro Artists to experience the Monopoly, water painting, graffiti picture book, and so on. Additionally, we also introduced the IGM competition and synthetic biology to them. Students had a great time here, and we hoped that they could participate in next year's IGM.
To this date, we have accomplished the basic work of parts engineering and preliminary society investigation. In the future, we will further improve our parts and hardware, make our project more accessible in reality, and do more human practices work. This is the general introduction of our project. For more details, please check the wiki page. Thanks for your attention and suggestions. We are the team GXU China.